So it looks like we have a pretty great crowd out there. So I'm going to start our presentations just to make sure that everyone has enough time to present their wonderful research. My name is Joshua Dullahan, and my pronouns are he and him, and I am the Assistant Dean of Instruction and Student Affairs at Hauser College of Natural Resources. I'd like to welcome you all to the Fall 2020 virtual poster session. Before we get started, I would like to recognize that Berkeley sits on the territory of the Huichin, the ancestral and unceded land of the Chuqueño Ohlone, the successors of the historic and sovereign Verona Band of Alameda County. This land was and continues to be of great importance to the Ohlone people, and we recognize that every member of the Berkeley community has and continues to benefit from the use and occupation of this land since the institution's founding in 1868. It's my hope that as we talk about the community that we hope to develop here at the College of Natural Resources and the research that we develop through projects like these, that is inclusive of the needs of the American, Indian, and Indigenous people. I know this has been quite an extraordinary semester for you as students, for your families, and for faculty and staff. I could definitely say that for staff. For the poster presenters, I want to congratulate them on completing your research. Research and teaching are core aspects of everything we do at the university. And to talk a little bit more about that, I'm going to hand things over to our Associate Dean, Dr. Lynn Hunsinger.
Thanks.
Lynn?
Yes, thanks, Joshua. I even managed to unmute myself on my own this time, so that's a sign of significant preparation. But you know, I think, as Joshua mentioned, right now is a time, an unusual time, a time when we've all had some opportunity to reflect on what it means to be isolated and a bit alone and not with other people and with as much as we'd really like to be. I know we all share that problem right now, as well as a lot of other things. So, I wanted to talk about research a little bit. This is bad for research because research is not something that we do alone. We need the stimulation, the insights, the help, and the company of our colleagues. You need to be involved. You as a person, you need to be involved. You need to be aware of the world in order to ask meaningful questions and questions that will make a difference. And gosh, we all look forward to when we can be together again safely. Research is about company. It's about collegiality. It's about flexibility and innovation and doing things over and over again to get it right. One of the singular characteristics is making mistakes, and you need your friends, your families, and your colleagues to support you through all this. Because making mistakes and starting over and following paths you could not have anticipated to find results that may surprise you or might even disappoint you, you need your family, you need your friends, you need your colleagues, you need students, you need teachers, you need all these people to help you through all this. You need to make so many decisions along the way when you engage in research. I imagine you noticed that, and that is not easy, making decisions like that. Science, ultimately, though, we don't have to be so serious. I mean, science really is a conversation. Sometimes it's an argument, hopefully a civil argument. It just demands that everyone puts their cards on the table, face up along the way. Oh, food. Okay, everybody has to put their cards face up along the way. You have to lay them right out on the table. And the evidence that you gather and you present and you share it with everybody to get their feedback and to reach out to them to share what you've learned. The evidence that you gather, that you present, it's always part of a larger question still. And that question is questions like, is there more to this? Does your work agree with mine? And what comes next in this area that we're exploring and we're so curious and we think it's important? So events like this showcase, they're part of that process of sharing. They're part of the conversation, the scientific conversation. Today, we're going to join that conversation during this session. We're joining, in fact, an ongoing conversation that's larger than any of us. We share our research, we invite others to teach us what they see and what we did, how we might do more, who we're reaching, and sometimes who we're not reaching. Let's share today in a spirit of accomplishment and even joy. I dare you to have joy today and to enjoy the togetherness we have today as we share our curiosity about the world. I'm looking forward to seeing your projects very, very much. Thanks. Patricia?
Thank you to, am I unmuted? Yes. Thank you to Deans Joshua and Lynn for starting us off. Welcome everyone to the Fall 2020 Hauser College of Natural Resources Virtual Research Showcase. My name is Patricia Hellier. I'm the advisor for the plant and microbial biology majors in the college. I'd like to introduce Craig Crossley, our office's intake advisor. You can see Craig down there. Together, we're going to be moderating today's virtual research showcase. So to that end, we'd like to go over the flow of today's showcase and provide some instructions. We're lucky to have 8 students presenting today. 5 are from the microbial biology major and 3 are molecular environmental biology students. Each student will have up to 10 minutes to present their research, including time for questions. If participants in the audience have a question, please use the Q&A button in Zoom to submit your question, and I will read it aloud to the presenter at the end of their presentation. Please note that the chat feature is disabled, so you will have to use the Q&A button to ask questions. That is it for the quick run over. We'll go ahead and get started with our presenters. Our first presenter is Shannon Chang, microbial biology major. Shannon?
Hi, I hope you guys can all hear me. Good morning. Today I'm going to be talking to you all about some research I've been doing in the Blackman lab on natural variation and developmental timing in sunflowers. Next slide. So some background on me. My name is Shannon Chang. I'm a first-year undergraduate in microbial biology and philosophy, and my research interests lie in plant biology, microbiology, and immunology. Next slide. So the past semester I've been working in Professor Blackman's lab, and the people there are doing a lot of research that I think is really interesting, so I'm going to share a little bit about that now. Next slide. So the lab as a whole focuses on understanding a couple of topics in developmental evolutionary ecology, including how developmental responses to changing environments evolve, the genetics of adaptation, development, evolution, and mechanisms of gene-environment interactions. There are 4 main projects, the first of which is on sunflower domestication. This project looks at alleles that contributed to domestication history and using ancient DNA to understand the origins of this domestication. The adaptation of developmental timing project focuses on how plasticity interacts with adaptation and the evolution of developmental responses as the range of a species expands. Gene family evolution looks into how gene duplications evolve new functions and novel traits. And finally, solar tracking project seeks to understand internal and external cues of growth coordination and mechanisms that govern solar tracking. Next slide. Okay, so that's a lot of projects. What do I personally focus on? Next slide. I'm involved in a project that seeks to understand the genetic and environmental underpinnings of floret maturation timing. You might be wondering what floret maturation is, and I'll show a video of that soon. Basically, sunflowers are made up of lots of little florets, which individually go through a flowering process every day, and this is what we call floret maturation. So what we want to understand is the environmental and genetic cues of late-stage floral development. We want to uncover the basis of floral attractiveness to pollinators. To achieve this, the group has used field studies, mutant analysis, time-lapse videos, quantitative genomics, and gene expression. Next slide. So if we're trying to understand genetic and environmental underpinnings of maturation, there must be some kind of variation to study. So maturation timing varies between domesticated lines, and this variation is thought to affect pollinator visitation to the plant. However, the genetic basis of floret maturation timing is unknown, and that is part of what we were studying. To do this, the group grew 3 replicates of 284 genotype domesticated sunflower lines, which is 1,000 plants. We took time-lapse videos of them in the field at UC Davis. This is last summer. And this is a step I'm involved in. We're scoring the videos for timing traits. We're using and will be using association mapping to understand the effects and correlations between SNPs or single nucleotide polymorphisms and environmental factors and timing traits. Pollinator scoring, or counting how many and what kinds of pollinators visit different plants, is currently in progress. Next slide. This is a video. It's an example of a time-lapse video that was taken in the field last year. And you can see what I mean by floret maturation. And play it again. And I'll point out this relevant steps in the next few slides. Next slide. Okay. So my work involves the step of scoring these videos for 3 timing traits: bud break, which is start of floral development; pollen star, the end of anther elongation and thus male fertility; and style elongation or stigma curling, which is the beginning of female receptivity to pollen. Next. So this is an example of bud break. It's when the florets begin to open and you can see the little holes and cracks in the smaller picture. Next. This is pollen star. They look like little stars in the tips of the anthers as they pop out, just like the earlier video showed. The yellow things on the tips. And next. Okay, and finally, this is style elongation, where you can see the styles opening out. And the smaller picture is a picture of a completed style elongation where they're fully expanded. Okay, next slide. Okay, so all of this data is input into a massive spreadsheet where we record the plant number, replicate, the date the video was taken, the direction the plant is facing, which will become important in a moment, the pseudo whorl, which are these little circles of florets around the flower, which all mature on the same day. They go outside to inside over a period of 7 days. We also note the time of first bud break, the time of 50% of buds have broken, the time of the last bud breaks, and the same is done for pollen star and style elongation. We also note confidence so that other scorers can go back and check our work, our initials, and notes of any strange occurrences that we observed. Next. Okay, so this is some preliminary data that was analyzed by one of our group members. The diagram on the left, beta is the slope of the line and is measured in units of minutes per day. So it's basically how many minutes later the maturation trait occurs each day. If you remember, we're studying timing. As you can see, maturation happens later in the day as winter approaches. As dawn happens later and later, maturation also happens later and later. On the right is the temperature data for these months. Okay, next slide. Okay, so floret maturation is associated with temperature. And basically, all you need to get from the slide is that bud break shows no correlation, and pollen star and style elongation happen earlier as temperatures increase. So increased temperature is correlated with earlier maturation. Next slide. So each of these flowers was filmed while facing east and while facing west. They were all planted in buckets in a field so that they could be rotated to face the east and the west. Basically, the big difference is that the east is warmer, while when they're facing the west, they're colder. And it's important to note that the common conception that some flowers track the sun while they're opened is false. They track the sun while they're unmatured, but once matured and open, they don't do that anymore. They stay the way they're facing. So since the sun rises in the east, the flowers facing the east have warmer florets in the morning, warmer than those facing west. As in the previous slide, warmer temperatures indicate earlier floret maturation. So flowers facing the east mature earlier than those facing the west. So these studies were done in order to study the effects of temperature within an individual flower without the added variables of waiting for a natural temperature change through the seasons. This allowed us to study the effect of temperature differentials without the other environmental changes that come with the seasons. Next slide. So that was the first part of the study, which is looking at how temperature and the way the plant faces affects maturation timing. The second part looks at pollinator visitation. The sunflowers, like many other important agricultural crops, have pollinating insects as their main pollination source. These insects are not all honeybees as commonly believed, and actually 40% of the pollinators are native insects. The hypothesis for the study is that pollinator visitation is more frequent in flowers that face east because they are warmer, they mature earlier, and have increased nectar or other rewards for pollinators. One study by another lab found that when west-facing flowers were treated with heat lamps, they matured earlier and had more pollinator visitation. This suggests that it is warmth and its effects that affect visitation. In order to make more conclusions, we're continuing to work on this huge data set, genetic analyses, and scoring for pollinators. Next slide. So finally, what does this research look like weekly for me? Next slide. So I have 2 lab meetings per week, one on Monday and one on Thursday. We go to a seminar once a week to learn about more interesting research going on at Berkeley and other places. And meanwhile, I score 6 to 9 videos a week, each containing 3 to 6 days. And these are the time-lapse videos that we saw an example of. And I make observations and interesting findings from those videos. Next slide. So that's all I have. Thank you for listening. I'll take any questions.
Thanks, Shannon. I think we had one question and it was why are CNR students so awesome? So that was a direct reflection of you and your research. So thank you very much. If Shannon's got her contact information there. If you have a follow-up question or if you want to submit one now, we have a minute or so to go ahead and do that. So we'll give folks a little bit of time to submit a question if they do.
I can't write in the box, but I wanted to ask how Shannon thinks this might affect agricultural production, if there's implications for that beyond what she talked about.
I think what I know about this is that like increasing pollinator visitation would help agriculture because I mean, getting more pollination is better for crops, but I don't know a lot about that, but I assume that it would help agriculture.
Okay, thank you, Shannon. We are going to go ahead and move on to our next presenter, Jessica Ma, who is also a microbial biology student. Go ahead, Jessica.
Okay, thank you, Patricia. Hello, everybody. Thank you for having me. My name is Jessica Ma, and I'll be presenting on my senior thesis project, which is on understanding the mechanism of ERAAP downregulation during MCMV infection. Next slide. So first, I want to give some background that is important for understanding my project, which is what is ERAAP? ERAAP is a protein that resides in the endoplasmic reticulum of the cell, and it stands for ER aminopeptidase associated with antigen processing. So what it does is it trims peptides so that they can be loaded onto MHC complexes to be presented on the surface of the cell so that immune cells can recognize these complexes. And this makes ERAAP a really good target for viruses because it is responsible for maintaining immune surveillance. Next slide. So cytomegalovirus is one of those viruses. It is a beta herpes virus, and it is the leading cause of congenital deafness. Next. And research has shown that HCMV downregulates the host protein ERAAP1. And my graduate student mentor has also seen that this happens with MCMV, where it downregulates ERAAP in mice. Next. So downregulating ERAAP is an immune evasion strategy because then HCMV or MCMV infected cells cannot present viral peptides to immune cells like cytotoxic T lymphocytes. Next slide. So my goal is to understand this mechanism of ERAAP downregulation by MCMV. And our lab has several hypotheses for how this occurs, and I'll be going more into depth in another slide. And understanding this mechanism is important because it can potentially lead to new drug targets to fight CMV infection. Next slide. So my graduate student mentor identified an important interactor of ERAAP through a lentiviral CRISPR screen. And the interactor is a protein called ERP44. ERP44 is a ER resident chaperone. It retains ER enzymes that lack the ER retention motif called KDEL. And one of those enzymes is ERAAP. And there's another one called ERO1 alpha, which I will also be talking about in my results. And The interesting thing about ERP44 is it exploits the ER-Golgi pH gradient to bind clients in the acidic Golgi and release them in the neutral ER environment. Next slide. So my two hypotheses are that MCMV downregulates ERAAP by either targeting ERP44 directly, which will prevent ERAAP retention, or MCMV alters the pH or oxidation state of the Golgi and the ER, which will prevent ERP44 from binding to ERAAP because, as I said, ERP44 exploits the ER-Golgi pH gradient. And that could lead to ERAAP secretion. Next. So how am I going to prove these hypotheses? Well, first, I measured the ERAAP, ER01 alpha, and ERP44 protein levels in MCMV infected cells. And I will also be measuring the pH and oxidation states in MCMV infected cells. And I'll do this by transducing pH and redox GFP probes into cells to stably express them in the Golgi and ER so that I can measure the pH and oxidation states. Next. So this is just an experimental setup of what I did to measure the protein levels of those three proteins. I first infected B6 fibroblasts with MCMV GFP for 32 hours, and I sorted the GFP positive and negative cells. The GFP positive cells are the infected cells, supposedly. And then I ran a Western blot for those three proteins. Next slide. So these are my preliminary results. On the right is just showing that I sorted for my GFP positive population. And on the left is my FACS plot. On the right are my blots for the three proteins. My ERAAP levels did decrease in the GFP positive cells. Those are the infected cells, compared to the GFP negative protein level. But the uninfected band for ERAAP is strange. It should be the same level as the GFP negative, but it's lower for some reason. And that is not a result that my graduate student mentor has seen in the past. So because of that, it is very a little bit difficult for me to make any conclusions about these results as a whole. But if we just look at the GFP negative, there is a downregulation in the uninfected cells for ERAAP level. And for ERP44, there is also a downregulation in the infected cells, which is pretty exciting because so maybe MCMV is actually targeting ERP44. And then if we look at ER01 alpha, there's actually an increase in the infected cells, which potentially there could be an accumulation of ER01 alpha. But in the end, these results are kind of inconclusive because of the strange bands in the uninfected protein levels. So I'll have to repeat this experiment. Next slide. So just to conclude, my project is determining whether MCMV downregulates ERAAP by interfering with the interaction between ERAAP and ERP44. And this could be due to either viral factors targeting ERP44 directly or MCMV altering the pH and redox state during infection. And so far, my results have shown that ERAAP and ERP44 levels may be decreasing during MCMV infection, while ER01 alpha levels may be increasing. Next slide. These are just my references. Next slide. And I would like to thank members of the Shastri Lab from Johns Hopkins University and Robi Lab in the MCB department here at Berkeley. And in particular, my graduate student mentor, Christy Geiger, who has helped me from the beginning, from developing this project and understanding, and also helping me with my experiments, and also my PI, Laurent Coscoy, who has guided me from the beginning as well. So thank you so much for listening to my presentation, and I'll be happy to take any questions.
So if you have questions, please submit into the Q&A button. We'll allow a minute for that. Okay, folks, thank you very much, Jessica.
Thank you so much.
Absolutely. I don't know what I did, but you are welcome. Next up, we have Rachel, who is also a microbiology major and the peer advisor for microbiology students. Take it away, Rachel.
Okay. Hello, everyone. So today I'm going to be presenting my research on assessing the role or effect of motility gene expression in tomato phyllosphere survival and disease protection. Next slide, please. So just a little bit of background. The reason why I'm doing this research is because we want to be able to help increase yield of tomatoes as an eventual application into agriculture, right? So in order to do that, we recognize that microbial communities are essential to the survival of their respective hosts. For plants specifically, there's two main microbial communities that we study. You have the rhizosphere or soil microbiome, which is mostly composed of microbes that fix nitrogen and acquire nutrients for the plant. And then you have the phyllosphere microbiome, which represents the microbes living on the aerial surfaces of the plant. That means on the stem, on the leaf, on the flower, any of those places count as the phyllosphere. So the phyllosphere microbiome is mostly composed of microorganisms which can help protect the plant against disease, although that's not their primary role. So the key question here is, how much do phyllosphere bacteria rely on motility and adhesion for their survival through wind precipitation and desiccation, which is the drying out of a plant and its microbes? And answering this question will basically help us think about how to use more effective biocontrol methods through possibly microbes instead of harmful chemical pesticides. So to continue, we have seen that express motility has not been shown in phyllosphere bacteria up to this point. And what that means is that we haven't really seen proof of these bacteria necessarily moving in the phyllosphere. It could be that they just land on there and sit and use most of their energy to just stay in whatever position they are. And this is potentially because production of flagella, you can think of these tiny squiggly arms that facilitate motility. These things are costly for microbes to produce. So it has been suspected in previous studies that phyllosphere commensal bacteria, commensal means bacteria that don't harm the plant, suppress an epitope called FLG22, which triggers plant immune response. So they suppress this to not trigger the plant immune response. And a different paper has discussed how phyllosphere commensal bacteria may actually express FLG22 in contrast to purposely trigger the plant immune system. So you see there's kind of a conflict in the existing research, right? There's one paper that's like, oh, it suppresses this gene, and another paper that says, oh, maybe it doesn't suppress this gene. One thing we do know for sure, though, is that phyllosphere pathogens, so microbes that harm the plant, have been repeatedly shown to express motility genes after producing a biosurfactant, which is kind of a lubricant. However, flagellar genes in commensal bacteria have not been manipulated experimentally, as I've seen, to confirm the effects on phyllosphere survival. Next slide, please. So the objectives of my research are to determine the importance of flagellar gene expression in tomato phyllosphere commensal bacteria on the tomato leaf, and also determine if lack of flagellar gene expression in tomato phyllosphere bacteria results in increased severity, disease severity incited by coexisting pathogens on tomato leaves. So if you look at the pictures below, this is a very common disease called bacterial speck. And in the Costello lab, which I do research in, we commonly work with the pathogen Pseudomonas syringae, which is the bacterium responsible for this bacterial speck. Next slide, please. So my hypothesis is that if expression of flagellant or flagellar genes is important in the phyllosphere, I would expect to see mutant commensal bacteria display decreased fitness in planta. So that means bacteria that I have purposely made to not be able to move, I would expect them to display decreased fitness on the plant. While if motility gene expression is not important, I would expect to see mutants display equivalent fitness to wild type or untouched commensal bacteria on the plant. And I would also expect disease severity to be increased in the latter scenario if flagellant expression by commensals is important to plant priming and defense against pathogens. So here are my methods. So the first thing I would need to do is to transform the lambda red recombinase system into a commensal in the lab that we have called Pseudomonas astruensis. Now the lambda red recombinase system actually comes from a bacteriophage, which is a virus that infects bacteria. And what this system does is it's able to cut DNA away and allow you to replace that DNA with a different gene. So on the left, you see our good old wild type Pseudomonas astruensis, which is a commensal bacterium. And I've highlighted the FLE-A gene, which is the gene that I would want to knock out, because if you knock out this gene, you can knock out motility, or a bacterium's ability to move. On the right, you see that the end result of this first step is to have the PUCP18 red S plasmid transformed into Pseudomonas astruensis. And this plasmid is the plasmid that encodes the lambda red recombinase system. And that lightning bolt in the middle just signifies the method that we use to do this, which is called electroporation. So after I make my cells electrocompetent, meaning after I make them able to be shocked without being killed, then I would shock this bacteria so that it can accept foreign DNA. And that's how I would be able to get the plasmid in there. Next slide, please. So the second step is to transform the kanamycin resistance cassette into Pseudomonas astruensis. So after I have shunted the plasmid, the lambda recombinase system plasmid, into Pseudomonas astruensis on the left, then I would do another round of electroporation to introduce another plasmid in there that encodes for kanamycin resistance. Kanamycin is an antibiotic, and the reason why we want to get this in there is so that in the end, we can have FLE-A be replaced by this kanamycin resistance gene. That way I can just put kanamycin into my media, onto my plate, and then this mutant bacteria can now grow because it can be resistant to this antibiotic. Next slide. So the last two steps are to transform FLE-A primers, and I didn't picture this step here, but it would be another step of electroporation. So I would shock this bacteria in order to introduce primers for the gene FLE-A, and this is how the lambda red recombinase system recognizes, oh, this is where I'm going to cut, and this is where I'm going to replace FLE-A with kanamycin resistance. So I'm going to do that, and then I'm going to do something called curing the PUCP18 red S plasmid. So that means getting rid of it, which is pretty simple. Apparently, you just have to put it in some sucrose water, and that can kind of deactivate the plasmid. So in the end, here you see that the lambda recombinase system does its work and successfully replaces the FLE-A gene with the kanamycin resistance gene, and then I can be able to select this mutant, this FLE-A knockout mutant on kanamycin containing media, because all other wild bacteria would die on this plate. Yeah, next slide. Cool. So after I construct this mutant, I would hopefully challenge this mutant against Pseudomonas syringae in vitro and in planta. So on the plant, I would do a spray inoculation onto 3 to 4 week old tomato plants, as you see kind of pictured on the bottom right, and hopefully a fight would commence against this mutated commensal bacteria, and Pseudomonas syringae, our resident pathogen. So after spraying on these bacteria, I would wait for a day, 2 days, 3 days, and a week, and measure abundance of bacteria at each of these time points using digital droplet PCR, which is a technology that we use in the Koskela lab to measure bacterial abundance. Next slide. So because we had some issues with like shipping of materials over and stuff like that, I haven't been able to do too much, but what I have been able to do is attempt transformation of the land-direct recombinase system into our commensal Pseudomonas astruensis. And the PUCP18 red S plasmid was provided courtesy of Dr. Lawrence Rami at Harvard, so thank you to her. And we're using this plasmid in particular because in Dr. Rami's previous experience, she was able to transform the land-derived recombinase system into Pseudomonas aeruginosa, which is a common lung pathogen that is genetically modified like pretty much all the time. And the system was able to be used to knock out several genes successfully. So we were thinking this would be a good choice to use this plasmid on another pseudomonad. The PUC4K plasmid was also provided courtesy of Dr. Rami, so thanks to her again. And I also made sure to use the Pseudomonas aeruginosa electrocompetence method by their lab as well. So basically what that means is in order to make different species of bacteria be able to receive that electric shock safely, there are different procedures that you have to use for different genuses and different species. So the ones that we use on Pseudomonads are going to be different than the methods we use on E. coli, for example. So in the rare case that this doesn't work, I have some alternatives in mind. So if I really can't mutate this commensal in the end, then I would do the same thing in E. coli and Pseudomonas aeruginosa and kind of see how they perform as phyllosphere bacteria. So we would be able to see as already established bad performers in the phyllosphere if they do even worse without motility capabilities. Next slide, please. Yeah, so that is the end of my research presentation, and I wanted to acknowledge a lot of people actually. So firstly, Dr. Stephen Lindau and his lab have been super helpful with teaching me these molecular methods, because often in the Koskela lab, we don't really use molecular methods. So I'm really grateful to learn lots of new things this semester and next semester. And of course, I would have to thank Dr. Britt Koskela and my graduate student mentor Elijah Melferber and the rest of her lab as well for mentoring me for the past 3 years. And of course, I'd like to thank Dr. Lawrence Rami and their lab for the plasmids that they sent over, as well as the PMB department, IB department, and Harvard Department of Microbiome and Molecular Genetics. And then last but definitely not least, I would like to thank the Hauser College sponsored program for undergraduate research for providing the funding for this project. So yeah, the next slide is just my references, but I will be open to taking questions now if anyone has any. Thank you.
So again, if you have a question, please submit it using the Q&A button in the middle of your screen. We'll allow a bit of time for that.
Amazing. While questions come in, Rachel, one question for you. How has your research process, I guess, changed because of COVID? And what would you have been doing differently had it been a normal semester working with this?
Yeah, so I would say during COVID, the biggest issue for me was one, learning new things like mostly on my own, and two, which I guess goes hand in hand, is like the what's it called? The like space requirements that labs have. So we can only have 2 people in the lab at a time in the Koskela labs, and oftentimes graduate students fill those spots first, which makes sense. So I have to kind of really, really be good at scheduling ahead of time when I go into the lab, make sure what I do what I do in the lab is like very intentional, so I don't just like waste a slot. So I would say if this weren't COVID, I would be able to be in the lab a lot more, get more hands-on guidance from my graduate student mentors and possibly other graduate students in different departments if I needed to reach out to them. But yeah, I would say that this has been a really, really great learning experience, and I believe kind of priming experience for graduate school, because my graduate student mentor Eli was telling me that like, oh, like you're basically getting thrown into the graduate school experience right now, which is like doing things pretty independently, except I'm also handling a full course load. So yeah, it's been a really good learning experience, although it was difficult. Lelania asks, if the bacteria do not grow on the tomato leaves, are there any alternative explanations for why that would be other than your proposed hypothesis? Oh, that's a good question. So if the bacteria, I'm guessing you mean the mutants, don't grow on the tomato leaves, I would say that that makes motility at least important, like motility genes to, I would say that would make motility genes, sorry, I would conclude that motility genes are important for commensal bacteria to have in order to survive on the tomato plant. Because in my head, I'm predicting at least some growth. I'm not necessarily predicting like 0 growth, although if that did happen, I feel like it would make my life easier in terms of concluding things, because I am only knocking out one gene that affects one function. So yeah, I would say that's my answer. Thank you.
Thanks very much, Rachel. We will go ahead and ask William to take the stage. William Sharpless is a microbial biology major.
Hi, how's it going? Again, my name's Will. I am actually taking an extra semester currently. I'm finishing a major in math as well as a major in microbiology. As you might be able to tell from my talk today, I like math a lot, particularly in the context of biology. I'm super glad Rachel's talk was before mine, because mine will also be about microbiomes, particularly agricultural microbiomes, and how we might control them through model-based methods. Oh, sorry, thank you. Next slide. So this is a pretty hot topic right now. This is actually, these are both graphics from a roadmap that was published last month nationally from something called the Engineering Biology Research Consortium. And they're trying to group this this mission that many researchers have in engineering the microbiome, particularly in 3 avenues, in distributing metabolisms, particularly for like synthesizing molecules or breaking down wastes, or organizing spatial control of microbes to try to do some sort of organizational structuring. And what I'll be talking about today is controlling diversity of sets of communities, and what kind of phenotypes you can induce by trying to control certain sets, like within your gut or within an agricultural plot, for example. And also, if at any point you have a super basic question, like, please let me know. I often take for granted like how long it took me to learn these topics. I was actually practicing this talk last night with my girlfriend, and I got like about halfway through, and she was like, so what is a microbiome? And just please, like anything like that. Again, this is the study of microbial communities, which have been largely intractable for the last 100 years because of how complex they are. There's many species battling at one time, and we don't necessarily know how to predict them. Next slide, please. So particularly what I'll be talking about today is enhancing the growth of rice, which is, I work in Adam Arkin's lab, who's a bioengineering professor here. And this is under a large NASA grant called the NASA CUBES mission. CUBES being the Center for the Utilization of Biological Engineering in Space. And this is within the Food and Pharmaceutical Synthesis Division. And our goal is to design a community for the rhizosphere, a controlled community, which is, a limited amount of members that you would then put into a hydroponic system to boost your growth of rice by protecting it from pathogens, by obtaining higher yields and such. And so particularly in regard to Rachel's talk, I will actually be talking about the rhizosphere, so the root associated microbiomes. If you go to the next slide, please. So the way that we predict microbiomes is very mathematical. This is using something called the generalized Lotka-Volterra model, which most people have actually seen before. If you've ever seen the periodic graphs of deer and wolves, how they, you'll see the deer populations go up and then the wolves do, that's actually a 2-member community. And so you can actually extend this model, as Lotka did, I think 100 years ago, to a species or a community of N members. And so here, I've written a differential equation for one of the members where I use the X dot symbol to represent the change in growth of that species. And the way the model works is it's in relation to the growth rate of the species innately, independently how the species grows. And then it's affected by the size of the populations of other species in the community, as well as how the interaction between those other species affects your species of interest. For example, you may have antagonistic members who are trying to suppress one species growth, and you may have cooperative members which try to boost each other because they share some metabolism. But unlike some microbiome studies, I'm not going to talk about any of those strategies specifically. I'm instead just going to talk about the overall community dynamics through models like these, where we, for better or worse, abstract away all of those strategies themselves. Particularly today, though, I'll be talking about how, as a spoiler, how I think the interactions which we model as constants could really be modeled as functions of the environment. If you go to the next slide, please. To start with an example, though, just so you kind of get what I'm talking about, I know a lot of people don't see these differential equations all the time. This is a 3-member community I've drawn here, where you can see the green arrows between the network there represent cooperative interactions, and the red inhibitory blocking arrows show what would be antagonistic interactions. And this is just a motif of interest, actually, but I've written out the system of differential equations here on the left. If you press next once. And this is the vector form of that, which is just a way to condense all of that writing. And you'll see me throughout the rest of this talk write or reference these kinds of vectors. If you go to the next slide, please. So again, my hypothesis is particularly about how these interactions and growth rates might be fluctuating with respect to the environment. I think most biologists would find this particularly obvious, honestly. This is for example, if you think about animals or humans or even microbes in this case as well, oftentimes they employ different strategies when faced with different obstacles. So if, your glucose or your carbon source is limiting in the environment, you may start to produce antibiotics, as some soil and fungal organisms do, particularly to try to then win despite what's going on. So I'm proposing one example of this here on the left in a model that I just kind of drew up, in which you'd say that maybe this depends on some vector C of certain metabolite concentrations within the environment, particularly like glucose or iron or maybe more toxic compounds, and they would probably degrade with relation to the species, or maybe they would be produced. And the point is that the interactions are probably functions of those concentrations. If you press next once, please. And so the point is, the reason we want to know this is because you may be able to predict how the community dynamics are going to change, as in how the diversity and how certain dominating members are going to change as maybe certain metabolites or other environmental cues such as temperature, oxygen, the amount of water in the system affect which species are arising. Particularly, this has some implications for controlling the microbiome as well. If you go to the next slide, I'll talk on this. So to introduce you to a little bit of system dynamics, if you don't, when we talk about the equilibrium of a system, that is the fixed state at which the system is going to remain after some amount of time. And in the Lotka-Volterra system, that means a fixed diversity of the community. And so it's already been solved by Lotka 100 years ago, and I've written it here. It's any any state, any diversity which satisfies this linear algebra equation on the right will then yield an equilibrium, a diversity which will potentially remain constant should the trajectory arrive there. And my point is, they are functions of the interactions and the growth rates of the community, the point is if you can control, or maybe you don't have control, but maybe you know how the environment is going to vary, you may be able to predict how those equilibria are going to change as well. Furthermore, when we talk about controlling a community, we often use a lot of network and graph theory, but to leave that out of this talk today for simplicity, the point is when you want to decide how many members you need to control in a community to change the entire community diversity, like for example, which microbes you might need to give targeted antibiotics in your stomach. It depends on particularly the network topology, we call it, based on the orientation of the edges and their weights. And so the point is if you can change those by changing some environmental concentrations, like I propose here exactly, or not exactly, you might be able to change the topology and therefore change which organisms you might be able to control. And therefore a treatment or a change in the rhizosphere may be better suited if you were to prime it by, feeding an extra glucose, for example. Next slide. As just a kind of proof of concept, this is a community, a 12-member community, formerly parameterized by lab members in the Arkin group of the gut. And so what I've done here is just through simulation, I've changed 2 edges in various places, 2 edges meaning 2 different interactions to try to create this motif on the left, we call it, which is a sub-community within the community. And we did that because it's a motif of interest and we think it has certain implications, but if you'll go to the next slide. Do you mind going to the next slide? Oh, yeah. So this is, you'll see actually what happens. So what you're looking at is 4 different communities. The one on the upper left, there's 2 ways to visualize all 4 of them, but the one on the upper left is the original. And what you can see in the others and the alterations is we can see that oftentimes we see large changes in which species are dominating solely by changing 2 edges in the graph. So the point is if we saw more than 2, which is potentially likely, we may see huge changes in the dynamics, which could be good or bad, but it's probably within our domain of control. This is, so to be more specific, what you're looking at here on the right is, oh, sorry, do you mind going back to the GIF? Thank you. So the graphs on the right for each community is the sheer population of different species. Different colors represent different species. And on the left is another way we visualize active communities by depicting the interactions based on the size of the populations at a given time. And so you can see the sizes of the populations change and they interact different ecological pressures on each other. Next slide, please. So the way that you actually ascertain these parameters, or like you decide how one microbe is interacting with another, is through a lot of growth culturing. To a biologist in the room, this means that like I'm just growing these species, particularly pairwise, which is shown to prove the overall dynamics many, many times. So you take, I've decided to use a 7-member community because of size constraints, basically on how many I can fit in a plate in triplicate. And if you press next once, you can see on this plate layout in the upper right, I pairwise every single member 3 times. And then I also actually do at the top some full communities so that I can use it to validate my data later. And in each of these plates, I then fill it with about 1 ml of media. And so in each well, there's just 2 species or the same species with itself. And then you take sampling or you sample over a 2-day period, and then you repeat the process by passaging those same cells into a new plate. And if you press next one time, could you press next once? Oh, is it not coming? Thank you. So I take each of these, one of these plates is going to have some fixed amount of media in it, which has, or will go at 1 temperature as I chose. So I then replicated that same plate 11 times to do once at each different temperature, and then once with 5 different concentrations of glucose subbed in. And just as a tangent, the reason I'm doing glucose in a lot of these is because rice plants, particularly in their exudate, will release glucose when they're stressed because they know, or we hypothesize that it it encourages the growth of certain species that are beneficial, the commensal organisms, towards the plant itself. But so that is the front end. The back end of this process, so I've done everything I formally said, and I'm in the process of extracting the DNA. And the way it works is you extract the DNA from all of those samples. You do a barcoded PCR, so you can mix them all together such that each well basically gets a little bit of DNA barcode. And then you can combine all of them and use an Illumina sequencer, particularly a MiSeq, to then sample them. And then what I've been doing in the pandemic time before I could get into the lab is I adapted a bunch of the code for doing a bunch of the sequence processing from the barcode to basically to demultiplex all of these samples at one time. And then if you press next once more, what that spits out is relative abundances within each well and over different times. And so then you can use those multiplied with the OD taken at the time that the samples were taken to decide how much abundance each species had together. And then I wrote my own code to then parameterize what those interactions are based on that data. But this is still in progress. So this is everything I've done. I still am in the process of doing a lot of the back end work where I'm deciding carefully if this video works, this is me doing one transfer here, what it looks like. It's honestly a massive amount of work. I had planned to use a biomech liquid handling robot, but the robot is unfortunately in an LBL site, and LBL has just this month allowed undergraduates to get on. But I ended up doing it by hand because I was pretty interested in this, and I've still got some more work to do, but I'm pretty excited about this. What I'm trying to do right now while I'm waiting for some of the back-end processes to happen is to design a controller, something called a model predictive controller, which is used in system dynamics and in engineering to guide a system along a specific trajectory. So in the context of this problem, that means that I would install this, or I would write the code for this controller, which is going to tell you based on what diversity you have in your wells, what you should then transfer your community to, either in temperature or in glucose, to arrive at a fixed diversity that you want to desire something, maybe because it contains commensal organisms and includes pathogen organisms. But that's all the work I have today. If you'd press next. Thank you. I just want to say thank you a ton to particularly those of the Arkin Lab and in the CNR group. Patricia particularly has now seen me all 4 years become what I am now. And I just want to say thanks a ton to her and CNR for supporting me in total. This has been a super awesome opportunity, and I hope to get it towards a publication in the next month. Thank you.
Thanks, Will. And I have absolutely no doubt that will happen. It was a very engaging presentation. Thanks very much. Okay, next we have Sherry Zia, also microbial biology major.
Hi everyone, I'm Sherry, and hopefully you all can hear me. Today I will be talking about the genes that underlie variations in cryotolerance or cold tolerance in yeast, and more specifically I'm going to be focusing on one gene, HEM12, which is the basis for my project. Next please. So the big picture question in our lab is, how does evolution build traits over long time scales? However, this is difficult to investigate because with shorter time scales, the organisms of interest would likely still be the same species. So you would be able to cross the two parents to generate viable offspring, which you can observe genetic recombination and investigate the genetic basis of those traits. But with longer time scales, the species, the organisms of interest would have likely diverged into different species, as I have shown here with Darwin's finches, with the ancestral finch having diverged over time to form many different species, each with a different beak shape and size that can't really mate with each other. So our lab uses the reciprocal hemizygosity tests for these interspecies comparisons to find the basis of the trait differences between, okay, yeah, I'll be a little bit more quieter. So this reciprocal hemizygosity test for interspecies comparisons to essentially investigate the basis of the trait differences between the different species, as I will get into later on in the presentation. Next, please. So our lab's model organism is budding yeast. The scientific name is Saccharomyces cerevisiae. I have a picture down there of the yeast, and this yeast can be found all over the planet along with its relatives. And actually the species of the Saccharomyces genus are 20 million years diverged. So on the bottom right, I have our lab's model of two sister species that are more closely related. So our model has that these two species diverged from a distant ancestor that was cryotolerant or cold tolerant. And at some point in evolutionary time, there was a split, and these became S. paradoxus and S. cerevisiae. And then after this divergence, S. cerevisiae acquired a series of mutations that made it less cryotolerant, and it also became more thermotolerant. So we hypothesized that there was possibly a trade-off between these two. Next, please. So this depicts the cryotolerance difference, as I was mentioning before, about how S. cerevisiae is less cryotolerant than S. paradoxus. And this cryotolerance difference between these two sister species was discovered by former BREM lab member Julie Chang. And what she did was she grew these two species in the cold and compared their growth. And this graph that I have here is actually my verification of her data. So I used liquid culture growth over time as a measure of fitness. And I measured growth by taking the optical density or the absorbance of the cells at a certain wavelength of light. And I essentially grew the yeast in a cold room and took time points or optical density measurements over the first few days. And I only did this for the first few days, but you can already see that S. paradoxus has kind of an exponential growth, whereas S. cerevisiae just kind of slowly inches along. And if I would have continued this experiment, both of the growth curves would have eventually leveled out due to limiting nutrients in the media. And both of the curves would have assumed kind of like an S shape. But from here, you can already see that S. paradoxus grows better at colder temperatures than S. cerevisiae does, and it fares better. Next slide, please. But what our lab is really interested in is the molecular roots of this macroscopic temperature difference, or the genes that underlie this growth difference or temperature sensitivity difference. Next slide. So as I mentioned before, the reciprocal hemizygosity test is a method that's used to investigate trait difference between species, and it's a novel method that compares homologous genes in different species by generating an interspecies hybrid. So here I have the alleles of a gene, the same gene in S. paradoxus and S. cerevisiae, and basically you would mate these two yeasts to generate the interspecies hybrid down there that has one allele from the S. paradoxus parent and one from the S. cerevisiae parent. Next, please. And so A here, you could see that the function of the gene from S. cerevisiae, or the blue gene, was knocked out by inserting a transposon at that location, so that only the other allele from S. paradoxus is functional. And in order to verify the transposon insertion, you would sequence part of the transposon and part of also the region next to it. Next, please. So B here, basically the same thing, except the other way around. So in this case, the S. cerevisiae allele would be functional, with the S. paradoxus one being not functional. And the two yeasts that I have labeled as A and B are called reciprocal hemizygotes. And basically they're hemizygotes on the same background. So with each only having one functional allele, you can kind of compare the function or the functions of these two alleles at a certain temperature or a certain environment. And so far what we found is that A with the functional S. paradoxus allele grows well in the cold, but B with the functional S. cerevisiae allele does not grow well in the cold. And Julie Chang, the former BREM lab member, did this test over the entire genome, and she identified HEM12 as a possible gene underlying the cryotolerance of S. paradoxus. And basically HEM12 was what's known as a top hit. So there was the most significant difference in growth between the two reciprocal hemizygotes. And so the next step would be validation of HEM12. And this goes into my project, which is to validate the effect of this HEM12 gene in conferring cold tolerance to S paradoxes with regards to that of S. cerevisiae, the same gene over millions of years of evolution. Next, please. So basically, first we would use reciprocal hemizygosity to generate reciprocal hemizygotes of HEM12 specifically, with all of the other genes being held constant. So this would be done through this process called transformation. And there would also be a bacterial plasmid, as you can see on the top left, with a cassette. In this case, it's called CAN-R. So we would use polymerase chain reaction to amplify this plasmid with the cassette so that there'd be many copies of it. And then but there would also be a homologous region on either side of the CAN-R cassette that would match up with the homologous region on either side of the HEM12 allele. So this would induce the yeast to cut up and replace the functional HEM12 allele with the CAN-R cassette, so that this would effectively knock out the function of the HEM12, and this process would be done through homology directed repair. And because the cassette CAN-R also confers antibiotic resistance, these reciprocal hemizygotes can then be plated on G4-1A plates to select for antibiotic resistance, so you know which colonies this RH-seq process was actually successful in. So then we would essentially isolate genomic DNA from these reciprocal hemizygotes, use PCR to amplify the genomic DNA, and then sequence verify part of the cassette and also part of the neighboring region to verify that these reciprocal hemizygotes were generated. And this was actually where my project has come up to at this point. So then the next step would be to grow the hemizygotes at cold temperature and to generate growth curves, as I've shown before, and from the curves arrive at a conclusion about whether the S. paradoxus allele is beneficial in the cold, more beneficial in the cold than the S. cerevisiae allele for HEM12. Next, please. And I would like to thank my PI Rachel Brem for her guidance and support during my time at lab, and for helping me with the slides. And I would like to thank Faisal Al-Zaban for teaching me lab techniques and what I know how to do in the lab, and also for helping me with the slides. And I would like to thank the rest of the Brem lab for introducing me to research science. Thank you.
Thanks very much. It looks like I forgot to open up questions at the end of Will's presentation. So now we will take questions for both Sherry as well as Will's presentation, if there are any. So we'll go ahead and allow a little bit for folks to do that. And again, if you have a question, please submit it via the Q&A feature.
Question for Sherry. How did you find this research, or what made you particularly pursue this project?
I was introduced to this lab because I applied through SPUR, and I was interested in evolution and how the genetic basis of different phenotypes of organisms, and I was interested in how those are selected for over time, which is how I came into this lab, and which particularly is interested in long-term trait differences between species, which was what I thought was interesting, because within species, like I said before, you could just mate them, but with interspecies, with different species, it becomes more complicated, and there's still a lot of work to be done, which really fascinated me on uncovering how exactly these two species that are so long, that shared such a distant common ancestor, came about through evolution and the pressures that shaped the traits that they now have.
Amazing. Thank you so much.
Okay, folks, thanks very much. That concludes the microbial biology student presentations. We're going to go ahead and start we have 3 remaining presenters, all from the molecular environmental biology group, major. And our first presenter from MEB is Josh Hauser.
Hi, everyone. Thank you for joining today and for all the great presentations we've had. Yeah, my name is Josh Hauser. I'm in my 3rd year in CNR studying molecular environmental biology. And I research in the Almeida lab in SPUR, where we focus on the pathogenesis and ecology of plant diseases. But I'd like to start with looking at infectious disease more broadly. We know pathogens can infect all forms of life, including humans, animals, and plants, and they can do this in a lot of ways. And my lab, we focus on vector-borne transmission, but things like COVID-19 we know spread very differently. And understanding these transmission dynamics it's critical to understanding how we go about disease prevention. And with SARS-CoV-2, for example, understanding the role of facial masking can limit transmission, how that can limit transmission has proven extremely useful and continues to allow us to protect the vulnerable and ourselves, and potentially even limit the severity of illness. So the role of transmission dynamics in infectious diseases are just absolutely critical to managing them properly. And if we go to the next slide now. So in my lab, like I said, we focus on the pathogenesis and ecology of plant diseases. Specifically for my project, we looked at Xylella fastidiosa, which is a bacteria, and it's transmitted to plants by insects that are common to California agriculture. These include grapes, almonds, and olives, and really can have absolutely calamitous impacts on the California agricultural economy, where, an example for grapes, they develop Pierce's disease, which can leave crops unusable. And so that's kind of one of the main, one of the big importances of this work is its effect so close to home here in Berkeley, as well as just understanding vector transmission in general. So we actually are kind of expanding our work off of something that a colleague Cornara did, where he found that as dose of pathogen increases, so does the rate of positivity of transmission, but that wasn't the focus of the work. And we really wanted to pinpoint that and try to understand how transmission is affected by pathogen population. So if we could go to the next slide. To look at this, what we did was we took 2 different insects, Graphocephala atropuncata, which are blue-green sharpshooters, and meadow spittlebugs, Phalaenus spumarius, and we raised them on positive source plants with Xylella. And then that lasted 1 day for a 1-day acquisition access period, and then transferred them onto test plants of grape for 2 days of an inoculation access period. But that started 2, 4, 6, and 8 days after that 1-day acquisition period to allow for pathogen development. And then to analyze this, we cultured the petiole samples from the plants and looked at qPCR results from Phalaenus, the smaller bug, looked at their heads to try to get estimates of how much pathogen was in the mouth parts. If we go to the next slide now. So this is what we found. On the x-axis, you can see inoculation access days after that 1-day acquisition access period. And on the y-axis, you can see the proportion of positive test plants. So on the figure 1, we see Phalaenus, and we see this kind of U-shaped curve for transmission efficiency as the number of days after acquisition increased. And then for Graphocephala, statistically, it was an insignificant difference between transmission efficiencies. And then for figure 3, that was actually kind of the preliminary data we have on qPCR for measuring the population of Phalaenus in insect heads. But again, it's really preliminary and it's only using positive transmissions. But so I kind of just wanted to include this to show where we're going with it. So if we could go to the next slide. What I think some of the really important results here are that when we looked at Graphocephala in there, we saw the transmission efficiencies were much higher than for Phalaenus. And that's an important conclusion because Graphocephala can harbor a lot more pathogen in their mouth parts, which kind of points us to this idea that the amount of pathogen does affect transmission efficiency. For Phalaenus, we saw that U-shaped curve. And we can explain that actually with some work done by my PI, Rodrigo Almeida, back in 2006, where in this middle image here, you see an electron microscopy image. And what he did was took images of inside of the vector mouth part, where when a feeding event occurs on its positive test plant, the insect is taking in some of this pathogen. And when the pathogen goes in, it's kind of free flowing around in the mouth part. And that's when it's really easy to shut off and transmit to another plant. So if the feeding event on a positive plant occurs relatively soon before the feeding event on a negative plant occurs, it's really easy to shut off the pathogen. So that kind of explains why the transmission rate was kind of moderate. But after a few days, the pathogen actually starts to grow on the inside of the mouthpart walls. And when it does that, it's flush against the inside and it doesn't shut off easily. So that explains that drop in transmission. But after a certain threshold population is reached, the pathogen actually starts to grow in figure B, out kind of pointing radially into the center of the canal of the mouthpart. And when it does that, it has less surface area attached to the walls, as you can see, and it kind of can be shut off a lot more easily when it feeds. So that explains why there's kind of a skyrocketing increase in transmission rate that we saw in the figure 2, because the pathogen is more populated and more populous, and it sheds off a lot more easily. So what the tricky thing here is that those EM images actually were in Graphocephala, not Phalaenus. And Phalaenus was the U-shaped curve one. So the question then is, why didn't that show up in Graphocephala as well? And I think one of the really important conclusions here is related to species level variance. And that might seem like an obvious conclusion, but a lot of times when we think about vector-borne transmission, we don't necessarily think about how each vector is unique. And so Graphocephala feed very favorably in the wild on grapevine, while Phalaenus don't. And we can understand this impact on transmission efficiency as we look at a feeding event as a chance for transmission to occur. So if an insect feeds favorably on a plant, then it's going to have more feeding events and hence more chances for transmission. So, but if it doesn't feed favorably, then there are fewer chances. And as there are fewer chances for transmission, each individual chance matters a lot more. So we can see this almost as the plant-insect interaction becomes less important in the case of Phalaenus, where it feeds less favorably. The microbial component of how that pathogen exists in the mouthpart becomes a lot more important and can even overwhelm the effect of the pathogen dose for a successful transmission. So from this, we really start to see that these diverse dynamics of a vector-host pathogen complex are really common in a lot of insects, in insect-borne disease. I think an interesting example is looking at West Nile virus, where there are two main species that transmit West Nile, the Culex salinaris and Culex tarsalis, which are from the same genus, but have drastically different transmission efficiencies. So it's really hard to track, depending on where those vectors are, how transmission can occur. And I mean, if we're looking at these two insects, which both transmit Xylella, they have drastically different transmission efficiencies, but they're from different families. So really, it makes sense that it might not work so smoothly to compare their transmission rates. So I think a really important conclusion from this is kind of accepting this idea that transmission efficiencies aren't going to be the same between vectors. And this kind of goes back to this common flaw in vector transmission and our understanding of it, where we like to clump the vector into categories of the pathogen or the environment and not treat it as its own thing. There's in infectious disease, there's this disease triangle, which looks at the host, the pathogen, and the environment, and that's where the vector gets clumped in. But we really should be thinking about it as the vector in its own category. And it is that we do think of it that way, and it's becoming more common, but I think that's a really important distinction we have to make and hold ourselves to, because as things like the climate crisis impede on us, there are going to be a lot more pandemics. It's just inevitable. They are going to be released from permafrost, things like that, and they're going to be vectors that are traveling to new places, and they're going to be on new ranges with new hosts. Humans are going to be coming into more contact with wild animals as there's habitat degradation, and we just have to be prepared to understand that transmission efficiencies are going to be different depending on the vector. And moving forward with our research, we reran this experiment for Graphocephala in 2020. That was what we've been doing for the past year or so. And we're really looking forward to seeing if those results hold true to what we found. And also looking to use a different analysis method than qPCR for measuring the Xylella populations in the vector heads of Phalaenus, because since they harbor lower levels, qPCR might not be sensitive enough. So we're actually looking into using digital droplet PCR to hopefully get greater resolution. And then if you go to the next slide, I just to thank all of the people in the lab that have helped so much with this research, especially Dylan, my graduate student mentor. And if you have any questions, I'd be really happy to answer.
Thanks, Joshua. So, looks like we have a question for Josh. You mentioned COVID-19 transmission. Do you know how our understanding of those transmission dynamics have changed?
Yeah, so that's a great question. Thank you, Chidera. So I actually have started researching with Dr. Monica Gandhi at UCSF, looking at these factors, and our understanding of dynamics have changed a lot, where at first masking wasn't really, if you remember back in, I think March, maybe even April, we weren't really sure if masks were necessary, and then they became necessary because we found out it's airborne. And then now we're learning that masking it has so many benefits where it protects not only the CDC just published it too, that not only does it protect the community, it protects yourself, and potentially even limits severity of illness. So yeah, that's kind of where my understanding of transmission is right now, but that's a great question.
Thank you. We have a question from Cynthia. Are there any current methods or interventions to decrease transmission rates?
So that's a really good question too. So I'm actually not quite sure what methods we have out in the field to limit that. Yeah, I don't quite know, because I guess that, pesticides would maybe get rid of some of the insects from eating, but I don't know how that affects the plant's growth. That's a good question.
And final question from Alicia. You mentioned lots of factors and that you think feeding behavior is most likely. Are there other prominent ones you think it could be?
Yeah, there are actually a lot of potential factors. One of them we were looking at, and we still have to kind of go over the data, but morbidity, how morbidity has played a role in transmission is something that we're looking into. And like I said, the vectors themselves, the mouth parts, the fluid dynamics inside of the vectors is really a critical component to this. Graphocephala are larger, so they can harbor more of the pathogen. Yeah, those are some other really important ones. And again, I think that they probably all do intertwine. It's not really going to be just one factor. And I think that's kind of the important part in our approach to understanding transmission dynamics.
Sorry, a few more. I know I said last, but also, where were the insects collected from?
Yeah. So they were collected from Napa in Berkeley and up in Arcata up north.
And is there evidence if insects moving north in light of climate change of insects?
Yeah, so that's actually a project. I know that's the case for a lot of insects across the country that are transmitting disease. Like I believe, but with ours specifically, we're working on a few projects with that, looking at how overwintering affects disease or affects insects' abilities to like stay over in their development in the winters. But I'm not sure necessarily of studies that show the insects themselves moving up, but I think their ability to tolerate and for the pathogen to grow longer for different parts of the year is changing. That's for sure.
Thank you. And then Mahmoud wrote, proud to see a former student presenting. How did the mathematics you learned earlier played into your research? A great presentation.
Oh, thank you, Mrs. Mahmoud. Yeah, you were one of the best teachers I've ever had. I'm really happy to have a firm understanding of calculus and linear algebra from you.
And then I think this is final question, unless we get more. Can you tell us a little more about the differences between two insects used in the study? Such a cool presentation.
Thanks. Yeah, so like I went over a little bit, Graphocephala are the larger of the two, so their mouth parts can harbor a lot more of the pathogen, and also they feed more favorably on grapes than do Phalanus. So those are the two main factors that I can think of right now about kind of the differences between the two, but they both are known to transmit Xylella, and that's kind of why we chose those two.
Thanks, Josh. That was fantastic.
Thanks so much.
Yes, absolutely. And so now we have our next molecular environmental biology student, Meghan Nolan.
All right. Hi, everyone. My name is Megan, and my faculty mentor is Professor Kathleen Ryan. My project is investigating the HIP-BA3 toxin-antitoxin system in Coulobacter crescentus. And Coulobacter is a gram-negative non-pathogenic bacterium. And we're interested in studying toxin-antitoxin systems because they've been implicated in bacterial persistence. And persistence is a transient phenotypic state that allows bacteria to survive different environmental stresses, including antibiotic exposure. If you have a population of bacteria that are exposed to an antibiotic, a lethal dose of antibiotic, a small percentage of them are able to go into this transient non-growing state, which allows them to survive until the antibiotic is no longer in the system. And once the antibiotic is removed, the bacterial population can then reconstitute itself. And if this happens in a pathogen, let's say you get a cold or something and you have a bacterial infection and you take antibiotics, a small percentage are able to go in this persistent state. It then causes chronic recurring infections, and which can lead to repeated doses of antibiotics. And the more often that we use antibiotics, the less effective they are. And so one of the best studied mechanisms leading to persistence are toxin-antitoxin systems. And Craig, if you can zoom in on that first figure there. Yeah, if you just right click and zoom in. Oh, I guess it's not going to work. All right, well, we'll just go with this. So this bottom figure here, under introduction, you can see, so a toxin-antitoxin system consists of a toxic protein and its cognate antitoxin. The antitoxin binds to the toxin, and when this occurs, it's inactive and unable to act on its targets. Antitoxins also bind to the promoter and stop transcription of these genes. And so as long as the antitoxin is around, the toxin is inactive, unable to act on its targets, and the genes are not transcribed. However, antitoxins are easily degraded by environmental stresses or antibiotics. And so when the antitoxin is degraded, the toxin is free to act on its targets, and also the genes are transcribed. And so if you looked at this figure in the top middle here, you'll see that a former grad student in the Ryan lab did a lot of work looking at the 3 different toxin-antitoxin systems that Coulobacter has. And what he found was that the HIP-BA3 antitoxin system behaves different than other toxin systems. And so HIP-A is the toxin, and HIP-A3 is the toxin, HIP-B3 is the antitoxin. And what he found was that phosphate starvation is a specific signal that causes an increase in these genes. And so the model that we have is that phosphate starvation somehow activates a protease, which degrades HIP-B3, freeing HIP-A3 to act on cellular targets in stopping cell growth and contributing to persistence, while at the same time allowing transcription of these genes to occur. And so what I wanted to find out was, how does phosphate starvation contribute to this phenomenon? And the first thing we wanted to do was to determine if the Coulobacter's environmental phosphate sensing and uptake system is involved in the activation of the system. And so I generated several strains of Coulobacter missing different components of this system. And the PSTS protein is a solute binding protein, a periplasmic solute binding protein, which binds phosphate and transports it into the cell. And as long as phosphate is moving in through this system, everything is great. So as soon as phosphate stops moving in through the system, it stimulates the kinase activity of PHO-R, which then passes its phosphate onto the transcriptional regulator PHO-B. And PHO-B binds to genes, binds to DNA, and turns on a bunch of genes that are used to scavenge phosphate and survive and adapt to this low phosphate condition. And so because transcription of these genes occurs when HIP-B3 is not around, we can use expression of these genes as a proxy for HIP-A3 toxin activation. And we first thought to do this using a transcriptional reporter that contained the fluorescent protein N-cherry under the promoter of the HIP-BA3 system. And in this case, anytime that these genes are transcribed, we should see a fluorescent signal. And it's a little bit hard to see here, but you can see that in the wild type strain in the top right of these fluorescent, in the figure 3 right here, you see a fluorescence signal, even when the wild type strain has plenty of phosphate. And that doesn't match the previous results that we had. So it's possible that the M-cherry protein is too stable in order to measure transcription in the system. And so we turn next to RT-qPCR. And this allows us to measure transcript abundance of the mRNA produced. And if you look in the figure 4 here, in blue on the left, you see the wild type strain. And each of these strains was grown in 2 different conditions. We grew it in one millimolar phosphate, which is the high phosphate conditions, and 30 micromolar phosphate, which is low phosphate. And we see an increase in transcription of the HIP-BA3 genes in this conditions in the wild type, which matches what we saw before. And we see an increase in the transcription of these genes in the Delta PSTS strain. And this is what our hypothesis was that, in a strain of Coulobacter lacking PSTS, the solute binding protein, this makes the PHO-B constitutively active. So all of the genes for scavenging phosphate should be on. And we hypothesized that the PHO-B-R system is involved in activation of this. And so we expected to see transcription on all the time because this strain of Coulobacter behaves as if it starved for phosphate, regardless of the amount in the growth medium. We expected to see transcription off in the Delta PHO-B strain because we thought that PHO-B was upstream of HIP-A3 activation. And while we see the increase in transcription in the Delta PSTS strain, which supports our hypothesis, the results from the Delta PHO-B strain are a little bit confusing because we see this strain, the expression of these genes increase when Coulobacter is grown in high phosphate conditions, and we see a reduction, but in the low phosphate condition. And this is a little bit more challenging to interpret. It could mean that activation of the system occurs through different pathways in different nutrient systems, or it is possible that the Delta PHO-B strain, it's an inability to elicit the correct transcriptional response, somehow activates a stress response, which causes HIP-B3 to be degraded. We next decided to look a little bit further about the involvement of the PSTS protein in the system. And a few other labs have studied the effects of the PSTS deletion on Coulobacter morphology. And in these papers, they noted that the PSTS deletion strain grows a little bit slower, but no one had quantified this growth rate before. We hypothesized that the slow growth rate of this strain was due to a constitutively active HIP-A system. And so if this were the case, introducing the HIP-BA3 deletion into the PSTS deletion strain should cause a restoration in growth rate. And if you look in figure 5 in the top right over here, blue is the wild type strain and yellow is the Delta HIP-BA3 strain. So this strain lacks the entire toxin-antitoxin system. And those strains have a similar growth rate. And then next you see the orange line, which is the PSTS mutant, and it does in fact grow slower than wild type. However, the green line that you see is the double mutant, the HIP-BA3 PSTS mutant. And we do not see a restoration in growth rate in this strain. In fact, we see a drastically reduced growth rate. And this is actually very exciting because the PSTS strain transcriptionally responds as if it starved for phosphate, regardless of the amount of phosphate in the medium. And so the fact that we see this drastic reduction in the growth rate suggests that the HIP-A3 toxin plays a significant role in allowing Coulobacter to adapt to and survive in low phosphate conditions. And so this work is ongoing and there's a lot more that we're going to be looking into. But I want to acknowledge my faculty mentor, Professor Kathleen Ryan, for all of her guidance and support throughout this, and the other members of the Ryan Lab. And I also want to thank the Rouser College of Natural Resources sponsored projects for undergraduate research for funding this project.
Thanks, Megan. So we will take any questions in the Q&A feature if you have them, and we'll give folks a few seconds to type those up. right, thanks, Megan. So we will move on to our final presenter. If folks think of questions also, please feel free to still put into the Q&A we can ask after. But for now, we'll move on to Dan Willett, our final presenter and molecular environmental biology major. Go ahead, Dan.
Hi, everyone. Great presentations, everyone. So yeah, my name is Dan Willett. I'm currently a third year studying molecular environmental biology and geology, and my project focused on the effects of amendments on soil carbon and nitrogen in California rangelands. Next slide. So what I did was process and analyze, or I will be analyzing soils that were collected as part of a much larger research project conducted by the Working Lands Innovation Center and my lab, which is the Silver Lab. And so what this project is doing is putting amendments on soils and measuring carbon and nitrogen and different carbon dioxide and nitrous oxide emissions to see how these amendments affect biogeochemical cycling, and if applying these amendments could be a practical method for mitigating climate change. And so there are multiple sites within this research project. The site where the soils I've been working with were collected at the Sierra Foothills Research and Innovation Center. And if you could go to the next slide. This is in Browns Valley, California, and the soils were formed from basic metavolcanic bedrock. And originally the site was a blue oak forest, but it was cleared in the 1950s and 60s for grazing. And since then, there hasn't really been much management. And as you can see, this picture is obviously during the wet season where it's all green and beautiful. Next slide. And so the soil samples that I've been working from, if you can see on the image to the right, are from that first block. And as you can see, there are multiple plots there, and each have been treated with a different amendment type or a combination of amendment type. There's compost, compost and ground rock, compost, ground rock and biochar, ground rock and biochar. And so the soils I'm looking at were collected before the amendments and after the amendments. And the goal of the research is to see how carbon and nitrogen levels change. And so just to give a brief overview of what these amendments are and the research behind them, I'm going to talk a lot about the carbon cycle and why we think these amendments will be a good tactic for mitigating climate change. I'm not going to talk as much about nitrogen because it's a more complicated cycle and there's not that much time in this presentation. So next slide. So the soils and the carbon cycle are very tightly interlinked. Soils are actually the largest terrestrial reservoir for carbon, both organic and inorganic. So as I'm assuming most of us know, that photosynthesis is one of the ways that we take carbon dioxide out of the atmosphere. Plants take CO2 and make sugars, and when they grow and eventually die and lose their leaves, they fall onto the ground, onto the soil, where microbes eventually eat at the organic matter and respire those sugars back up as carbon dioxide. But not all of those, not all of that organic matter is actually decomposed. A lot of it stays in the soil as humus, which is organic, rich layer filled with nutrients, and this is resistant to further decay. So we actually see a net accumulation of about 3 gigatons of carbon worldwide in soils. And so because we know this cycle, there's a thought that, okay, maybe we can see if we can sequester carbon knowing this. Next slide. So unfortunately, since the Industrial Revolution, we've lost a lot of carbon from soils back into the atmosphere as CO2. This has come as a result of land use changes and soil erosion, just not great land management. And this is equated to about 12.5 to 25 parts per million of the current increase in carbon dioxide in the atmosphere. And with this, we want to ask the question, is there a way we can reduce these soil losses, or even a way to increase the amount of soil stored or sequestered by soils? And that's where we lead into our amendments. So compost and biochar are organic material that we've been adding to the soil. I feel like most of us maybe have heard of composting and putting them on different crops, and because it's a great fertilizer. One of the things that compost does, as I kind of described in the soil carbon cycle, is that when you add organic material to the soil, not all of it is respired, but a lot of it is held in the soil as humus or also in different soil structures, such as aggregates. And this increases soil organic carbon or stable carbon in the soils. And this is super important for soil health, as higher levels help drive better soil structure, nutrient cycling, water dynamics, microbial activity, and biodiversity. In addition, when you have higher soil organic carbon, that means that there's less atmospheric carbon available, which is lowering the greenhouse gas impact. Biochar is a similar process. It's not as fertile because it's mostly carbon. And for those of you that don't know, biochar is organic material that's been burned at low oxygen levels and at high heats. And it's a process called pyrolysis. And so another benefit of having high soil organic carbon is because the soil is so much healthier, it promotes more productivity of plants and increases inputs to the soil and further increases the amount of soil organic carbon, or at least that's what we think is happening. And there's been evidence of this. Next slide. So ground rock, as you might assume, is probably different because it's not made out of plants or plant material. And the way that ground rocks relates to the carbon cycle is through the overall geologic carbon cycle. So on long time scales, where carbon dioxide comes from is volcanism. And when it enters the atmosphere, we see a greenhouse gas warming effect, a greenhouse effect, and that causes evaporation and precipitation. And when that happens, when water comes down and interacts with rock, we actually see a reaction take place called chemical weathering, where two carbon dioxides are pulled out of the atmosphere. They're combined with water to create carbonic acid and interact with calcium silicate materials to create carbonate ions. And so we see carbonate ions develop in the soil, or that's what we're assuming is going to happen. And eventually these carbonate ions travel down to the ocean because they're water soluble and eventually precipitate as carbonate, either through like calcium shells is a great example of that. And you do lose carbon dioxide during this reaction, but the net gain or the net loss of carbon is you take 2 out from the atmosphere and you emit 1 when you make these shells. Next slide. Yeah, and so because of this process, we're assuming that when you put ground rock amendments on the soil, which is this gray pile on the right here, when it rains, you're actually pulling out carbon dioxide from the atmosphere and storing it as carbonate anions. And ground rock applications have actually been used for centuries. Gypsum was a really important, and I believe still is, amendment in California soils. It helps alleviate the acidity of soils and helps with retention of organic material, which again is good because that leads to increases in soil organic carbon. And the amendments that we're using are a mixture of pulverized gypsum, basalt, and wollastonite, all of which are highly weatherable, and we're hoping will help sequester a lot of carbon. Next slide. So what I've been doing in the lab so far, the soils were already collected for me. It would have been really cool to go out into the field and do that myself, but unfortunately, I was not a part of the lab a couple of years ago. But the first step is taking the soil and sieving it through a 2mm sieve, which you can see in the top image up here. It's kind of like a grate where you add the soils on top and kind of swish them around and grind them into the grate to get out to filter out the larger rock materials and larger grass particles that we wouldn't consider part of the soil. The next step is milling. And sorry, I forgot to take a picture when I was in lab last week before I submitted this presentation. But what you do is you add this sieved soil into little canisters and shake them up in this machine, which helps pulverize and homogenize the soil samples, which makes it a lot easier to analyze them for carbon and nitrogen using the elemental analyzer. And I'm still in the milling step right now. I've been processing, or I've gone through almost 200 soils, so it's been pretty time intensive. hoping to continue it on to next semester. And this bottom picture is just the little vials of the milled soils. And this was from a week ago, and there's a lot more filling this now, and there's still a lot more to go. And then, of course, after the elemental analyzer, I'll be doing data analysis and comparing the carbon and nitrogen levels before and after the amendments. Next slide. Yeah, and I'd like to give a great big thanks to the SPUR program and the alumni who provided funding and made this possible. It's been tough doing school online, but it's been really nice having an opportunity to come onto campus and still get somewhat of a college experience. Thanks to Wendy Silver, who really encouraged me to apply for this funding. Andrew Jones, the postdoc, who's doing a lot of work with the Working Lands Innovation Center. Summer Ahmed, the Silver Lab manager, who's always been there to answer questions and get me supplies. And of course, all the lab technicians at the Sierra Foothills Research and Innovation Center who collected all the soils and made this research possible. And the next slide just shows some references for people who might be more interested in learning about soil science, biogeochemical cycling in soils, and the different amendments. Thank you.
Thanks, Dan. So if folks have questions, please submit in the Q&A feature. While we're waiting for Dan's questions to come in, we'll go ahead and ask one from Lalenia, the MEB advisor, to Meghan, the previous presenter. She says, hi, Meghan, I may have missed this, but any thoughts on why you received the unexpected results with, I think it was PHOB, P-H-O-B, I'm not quite sure how to say that. So Meghan, if you have an answer.
Yeah. So PHOB is a transcriptional regulator that binds to DNA and turns on about 20 to 30 genes that are involved in allowing Coulobacter adapt to low phosphate conditions. And so it's possible that by lacking PHOB, there's some sort of one of those genes that are responsible for helping Coulobacter adapt to those conditions. It's possible that something not being turned on in the low phosphate conditions or even in the high phosphate conditions might cause a stress response. And this stress response, because we don't know all of the, we have in this figure, you can see there's a lot of question marks of what HIP-A3 phosphorylates and activates, and questions about what actually activates the protease that degrades HIP-B3. And so there could be different pathways converging in this. And so there's a lot of work for us to do to kind of uncover what exactly is happening there. I'm not sure if that answered your question entirely. Another possibility is that it could be possible that the HIP-A3 system is regulated by different pathways during different nutrient conditions. There could be something beyond phosphate starvation that we haven't looked at yet.
Thanks, Meghan. So we're going to still, sorry, big noise, my end. We're still going to allow a couple while I'm speaking here. If you have questions for Dan's presentation, please do submit using the Q&A. Oh, we have one here.
Oh, Lalenia just said thank you to Meghan.
Lalenia said thank you. So if you have questions, please submit, but I'm going to go ahead and start saying our thank yous and our wrap-ups. One more. Same one. So thank you to everyone who has presented. That concludes our Fall 2020 Virtual Research Showcase. We are really so impressed with all of your amazing research experiences. We want to thank the attendance. On a personal note, we want to, speaking for the advisors in the college and the staff in the college, we're really so proud of everyone. Well done, all of you. This presentation will be available on our Hauser College of Natural Resources YouTube page, and you can just Google that, search Hauser College of Natural Resources on YouTube, and that'll come up. And it's going to be available on our website as well. If you'd like to hear more research presentations, the SPUR Undergraduate Student Researcher Panel will feature Fall 20 SPUR students sharing their experiences as a sponsored student researchers with their faculty-initiated projects. And that's going to be next Wednesday, November 18th from 12 to 1 p.m. And as you can see here, Craig will talk to you a little bit about the virtual bulletin board.
Yeah. Hey, everybody. So like Patricia said, I have it pulled up here in front of you. And I guess Rouser College folks will know, in lieu of regular weekly newsletters, we've created this virtual bulletin board where we have everything from, the showcase in which you just attended, internships, job opportunities. We even have recordings from previous things. Down here, we have, faculty panel and some interviews with students, as well as the upcoming honor symposium information. We have everything from grad school call outs, event things, spring 21 courses. I send an update every Monday afternoon about this, but this is, you can access it at any time. We update it throughout the week. And so look for those emails Monday afternoons. I just highlight kind of what's new that week, but you can click the link at any time and see what else going on campus. It's just kind of our initiative to stay in touch and kind of have that back and forth interaction, since it is such a remote time and a strange time. But it's just been such a privilege to hear all the fantastic work that's still happening out there. Thank everybody for attending. I think it was amazing.